put it. Okay. So, Alan, thanks for coming and thanks for the introduction. I'll be talking. Wait, let me remove this panel. Hold on. Hide. You say float. Meeting control. Hide. Yeah. There you are. Okay. So we talk about hydrothermal carbonization, basically as a relatively new mean for wastewater treatment or for sludge treatment more than the wastewater itself. And yeah, in the next half an hour or so, a little bit more. Well, the slides are not moving. Next. There you are. God gracious. Okay, now it starts moving. So I decided to steal a couple of minutes just in case, although I know most everyone here but I don't know. I didn't know who is going to be on the Zoom. So I want to say that we're coming from the Sde campus of Gurion University, where we have 3 academic institutes, the Institute of Agriculture and Biotechnology, the Institute of the Water Institute, or the Zuckerberg Institute for Water Research, and the Institute for Dryland Environmental and Energy Research, and the Albert Katz School for Graduate Studies. And we're looking for students. So Binchas was here, said no students. We're in the pretty much in the same boat at times. And so if there are students out there, you're welcome to apply. Again, for not knowing who is out there, I would just say for 1 second that you know, water is obviously crucial, specifically in this area, and 70% of it today is used for agriculture, and it's expected to grow. And all the rest of the water usages are actually smaller. Municipal is about 10%, industrial is about 20%, and so on. So the issues of water and agriculture is obviously important, and here I should not really say too much of that. And again, as you know, but it's important for me to put a line, is to say that as population growth and agricultural activity is growing, waste the waste volumes and quantities are increasing dramatically over the years. And in terms of, which is the focus of this talk, the organic, wet organic waste, sludges, you know, from cow sheds, chicken manures, and all of these kind of agricultural wet organic waste. They have some, it's hard to say, but they have some general properties that we can define to some extent at least, and they surely need solutions and in order to treat it well or to reuse it. So these organic compounds are often with high moisture and poor dewatterability, and it often contains whether it's micropollutants or others, nutrients and so on. And while it's a waste, it's obviously it has at least a great potential for energy and nutrient recovery, and that's what I'm gonna talk about. So that's in terms of introduction. I'll have a little bit more, but towards the technology that we're using. I put this slide again for the same purpose, to say that there are billions of people who, it's not only agriculture, it's also people who are facing a sanitation issue, and billions in the world. This is example from India, and basically you can see that hundreds of millions still using open defecation. Anyway, to say that organic waste is a real issue, both in agriculture and in urban environment or rural environment in general. And that's the point I wanted to make. The other thing which I think it's really important, and probably few of you are familiar with that, is that there is a great potential, because today, in order to treat wastewater in general, we need about 1,200 to 2,000 and above, maybe 3,000 megajoules per 1,000 cubic meter, which is really pretty much the energy cost to treat wastewater, where in the wastewater itself, if I assume COD of 500 milligrams per liter or 500 grams per cubic meter, it's the same. It means that we have like 500 kilos of COD or organic matter, for the sake of this talk, per 1,000 cubic meter, which means that every day we produce, if we took a 1,000 cubic meter wastewater treatment, it means that we produce 500 kilos of COD, and each kilo of COD equals potentially 13 megajoules of energy, which means that we have about 6,500 megajoules per 1,000 cubic meter, and in the waste, where it takes us about third to treat it. You see, so basically we have enough energy in our waste to treat it and להישאר עם עודף, and to have excess energy for other purposes. And this resource is untapped, or it's not used for whatever reasons. I think people just didn't pay attention to it before, and today we all start to look at it and try to gain this back, and that's what it is all about. And again, is part, there are different treatments for sludge or wet organic waste. And I would say that probably anaerobic digestion is the most abundant one, where we anaerobically digest the waste and get biogas and digest a little bit or lose some of the solids, sometimes 50%, sometimes 70 or 80%. It depends what is the resource, but we still end up with a product with leftovers, I would say, or effluents that are often contaminated. And that's an issue with anaerobic digestion, although it's really abundant, specifically in decentralized solutions or on-site solutions. Then there is composting to sludge, and that's an aerobic process, and that's also contains quite a lot of faults. And I know people are working on it here, but Pinchas is not here. I think he's in the Zoom, but composting is better than not doing anything, but it's far from being an ideal solution. And then people use pyrolysis, and mainly dry pyrolysis to begin with. And the issue there was that when you heat this waste for energy resources, you really gain energy. But in order to dry the water before you start pyrolyzing your organic matter, you lose so much energy just for evaporating the water, for drying the matrix, that the energy balance of many of these processes is negative. So we treat the waste, we gain energy, but it's less than what it cost us to do it. And the last, and the one that I'll be talking is, I think it started to be used for treatment of organic waste somewhere in 2010, 2012, maybe 2 years earlier. But that's around this time that people started to look at it. carefully in terms of waste treatment, and it's a wet pyrolysis. And what it is, it's a thermochemical conversion of wet biomass. The temperature range is around 180 to 250 degrees. It's much lower than the dry pyrolysis, which is hundreds, 500 degrees and above to 1200 degrees. So it takes much less energy to use. And actually, we use the water as the active, reactive substrate. And the reaction time is from minutes to several hours. And there are two main chemical reactions that occur in there: dehydration and decarboxylation. And I'll be saying a few words about that. And really what happens, or I would say that what we're doing is we're taking our wet waste, we introduce it into a pressure cooker, and we heat it. That's all there is to this methodology. And we heat it to that it would get to around 200 degrees, anywhere 180 to 250 or so. And then what happens, the water is considered to be in a subcritical temperature, subcritical water. The Kw goes up. You look here, 200 degrees, and the Kw of the water actually goes up from 10 to the minus 13 to 10 to the minus 11 or so. And that means that we have much more hydrogen and hydroxy ions, acidic and basic reactions in this solution. We have polarity drop. The dielectric constant go from about 80, which is the normal water constant, to less than 40 at 200 degrees, which is like acetone. The water turns into solvent. Basically, the water from being like, O and 2H is becoming almost like a solvent, and they attack organic matter. And the density goes down as well. And what it happens, and I'm not going to go into that, but what at least not too much, what happens is that because of the reactivity of the water and basically the heat and the temp and the pressure, we gain lots of organic matter transformations. So lignins are affected at the higher temperature. Cellulose at above 200 degrees, that starts to break down. Hemicellulose and carbohydrates at lower temperatures. Remember, lower, it means above 180. In our case, and proteins. And it goes through, part of it is aromatization, and part of it, as we said, decarboxylation, and some is repolymerized, and eventually we end up with some sort of a solid component, which we term hydrochar, because it, as you will see very soon, it has a hydrochar-like properties. And it depends what we start with, because not every material gives the same product. And we end up with liquid phase, because we started wet. So the liquid phase, though, it's now sterile. And other things that happens there is some of the micropollutants are broken down into, are being degraded completely. Pharmaceuticals are being degraded completely, and so on. So, but we have a liquid phase which is usually rich in nutrients, and it's sterile, obviously no pathogens at this temperature. And we have some sort of a gas phase that is very, very small, because when we do it in a pressure cooker, we don't let the water evaporate. It stays in its water form. So the gas phase is really very, very small compared to any other methodology that I'm aware of. And what we get is mostly CO2, some methane and hydrogen. We get a little bit of ammonia, depends what we start with. We get a little bit of sulfides, and so on. So it's a complex process that a lot of work currently, or what we're trying to do is look at these cycles here. But that's again, I don't want to look at it too closely in this talk, but it's interesting. The other thing that we get is sometimes is HMF and furfurals and some polyphenols. So if we want to use these compounds for agriculture, at times it can be inhibiting or so on. And overall, just to put it in context, it's basically we're mimicking coal formation. The process is mimicking coal formation in nature, where wet organic waste is covered and under pressure and heat, and it takes 100,000 years, and you get coal, or million years, and you get the real coal. So you do that in a reactor in minutes. Now, after spending enough time, I guess, for introduction, I just want to show some of the things that we did, and I don't know, the insights that we have from using this technology or using this practice as a wet waste and sludge treatment, and discuss that. So basically, the experimental setup is fairly simple. We take different sources of wet waste. And when I say wet waste, it can be, we really need to choose what is the solid water ratio or the solid liquid ratio in this case. And we had quite a lot of research about that, because the more water you have, sometimes the process is better because you have more reactive substrate, but obviously you heat water. And if you heat water, you lose energy, because it's the same like with pyrolysis. So we found out, and I think we can go even better, that 1 to 3 solid solid liquid ratio would be the best. Will give the best results in terms of or optimized result between the energy you invest and the energy you recover. I think it can go even lower. Depends what resource. So you take wet waste, you put it in this pressure cooker, this reactor, you heat it to whatever temperature that you choose, anywhere in this range. You don't want to go higher, because then the water properties are changing, and again, you invest energy more than you may need. And then you end up, we said, with solid, with liquid, and with gas, which I'll ignore in this talk. So in the solid, if you dry it, it will look like hydrochar, it look like that. I brought you, I want to see that. We thought we were really clever. We call it pool of corpus, because this is the human excretion. And yeah, so it's char-like material. It's really hard to distinguish. And then the liquid, and then we tested the liquid for different usages. One of them was to grow plants on it as a liquid fertilizer. Again, so this is the experimental setup. And this is how this liquid look like. It's a bit brownish in color. The hydrochar is black in color. And the gas, I said, I'll ignore it, although it's an interesting feature on its own. Usually the temperature that we used were around 180 degrees, 210 degrees, and 240 degrees for different reaction times. We use these kind of reactors, so we heat it in an oil bath, basically, and after we got to the temperature inside the reactor, we let it stand for whatever minutes that we decided upon, and then we took it out and quenched it in an ice bath, so these are the reaction times. And generally speaking, what we found is that the energy demand is usually depends on what temperature and how much time would be anywhere between 17 to about 30% of the energy output. And we got this product. This is a larger pilot reactor to run some study, but to run these larger studies. But again, it's basically a pressure cooker. Then, before we started, what we realized, and that was done by different people in the past, that they try to they say that it's not only the temperature that really important in the process, but it's the temperature and the time, you know, and the reaction time. And they try to put some sort of an index that they call severity of reaction, and it was a function of the temperature and a function of time. And you can see that the temperature affects more than the time, but still. And what we did is we wanted to see if this severity factor can change, you know, when we hit it to 180 for 30 minutes, what it, you know, what it would be, or how we can compare it to 210 in an hour. Okay, and we use this severity equation, and we tested different severities, okay, according to this equation that is composed of different combination of different temperatures and different times, okay? So in order to get the same severity, if we hit something on 180 degrees, we need 120 minutes, where if we use 270 degrees, we need to use only 2 minutes, and so on. So we had 5 of these, and we look at the effect of severity. This slide run out. No, sorry for that. And I'll go back. I don't know how it came here. And what we found out, that if we look at the severity factor, and this is just a representative parameters, in this case, the calorific value, how much calories we get in the in our coal or hydrochar, then other than this outlier, you see all this group is the same severity that was done by the different temperature and time combination. And it's the same severity, and you can see that it doesn't matter. It's not the temperature, it's not the time, it's the combination of temperature and times that gives the properties of the hydrochar. And why is it important? Because we can now actually predict what is going to happen if we use severity. And in practice, why it is important, because to hit something to 180 degrees, it's a very different pressure than to hit something to 270 degrees. It's a different materials that we need to use, the reactor costs differently, and so on. So you have to optimize between time and temperature and what you have. Okay. What happens when we do that? Then we said decarboxylation and dehydration mainly. So what changes often is the oxygen to carbon ratio and the hydrogen to carbon ratio. And if this is the oxygen to carbon versus hydrogen to carbon ratio of a normal biomass, yeah, then as we heat it or as the severity increase, what will happen is that we'll take hydrogen and oxygens out of the molecule, out of the solid, and to a certain extent, we will produce CO2, but it's usually less than the dehydration. And at the end of the process, what happens is that the ratio of these ions is similar to coal. And that's called this Van Krevelen diagram, which identified different compounds like biomass, peat, and so on are based on their oxygen to carbon and hydrogen to carbon ratio. And if we look at the percent of the carbon, so if that's the original sludge, which had in this case something like 38% carbon, you see what happens. It gets to almost 60% carbon after this digestion. If we look at the calorific value, it gets from 15 megajoule per kilo to something like 24. And if it's human excretion, it gets also to almost to 30 megajoules. And that's really high quality coal in terms of energy. And the energy yield is, you see, around 70%, sometimes above. Here, I think we had some faulty results, but it's around 70 or 70 something percent. Even though if you look, it's harder, it's not always that clear to see, but the mass yield, you see, is going down as we heat more. So we get less mass, but the carbon is more dense, and therefore the energy yield stays pretty much constant. And there is always a net energy yield if we take into consideration how much we invested into heat it and dry it and so on. We also see that there is some percent of ash into this biomass, and this is really important because if the substrate contains a lot of ash, if it's a digested, for example, substrate, then the ash content is sometimes higher, and the energy content doesn't get the energy content here is not getting high enough, and then you cannot use it for energy purposes. Then what we did is we looked at the combustion properties of this hydrochar. So we said, okay, we want to use it as energy. Let's see that it can actually be used as energy, because often biochars and all that contain energy, but they're not very efficient in terms of energy use. So, and I'm not gonna go too deeply into the details, but what you can see is fairly similar combustion properties of hydrochar, which is the red one, of pyrochar, which is biochar that was done in dry pyrolysis, and subbituminous coal from a mine, okay, which is this graph here. And basically they're all the same. So the idea is that it can be replaced as a substrate directly into the current facilities, and you do not need to build any generators or power plants that are specific to this carbon.
But the temperature that you use is 200, not 5.
This is the burning properties. I use 200 degrees to, yes, this is the gravimetric, thermogravimetric analysis, and that demonstrate the production, the combustion properties. Okay, so it burns, the coal itself would burn mainly at around 400 to 600 degrees. Yeah, and then that's where all the processes will occur. And we also demonstrated, I'm not gonna, it's the same combustion properties. What I wanted to show that I can predict, or we can predict this, that if you tell me, or if, that if we burn or if we prepare the char at a certain severity, we would not be able to predict what would be the combustion property, because it changes according to the severity that this product is being used. So this gives us again a tool to optimize between the combustion properties, if we want to use this thing as an energy source, and for example, the liquid properties that remains if we want to use it as a, for example, as a liquid fertilizer. Okay, so that's what was done here. If we look at this char in terms of properties, what one would find out that these are very hydrophobic char-like material. The hydrophobic contact angle is, I mean, the contact angle is typically more than 70. Sometimes it's even higher than that. And you know, it changes the soil properties, which if it is amended to soil and not used as energy source. And why would we like to use it as soil amendment? First, it's another usage. And secondly, as I said, not all the char has high energy properties, because it depends on the amount of ash that we start with. So in this bit, I would just show few studies that we did of how can we use the hydrochar as a soil amendment rather than energy source. And what we did is basically we applied it at 10 to 40 tons per hectare as a soil amendment. And we grew plants. Typically, I like to grow lettuce because it's a good model plant, not because we eat it. It's a good model plant. It's sensitive. It's a short growth period, like of 30, 40 days, and it's not budding, or we finish the experiments before it buds, so it's only green leaves. And what we see is that when we compared slow release fertilizer to carbonized to this HTC or hydrothermal carbonization soil, the hydrochar soil amendment, we see pretty much, that's by eyes, the same results. And believe me, it came pretty much the same, which was surprising, I must say. We also looked at it as an absorbent material. So we activated this hydrochar with phenyton reaction. So we introduced iron and H2O2 and mixed it together. And we had this, actually, I thought I had a slide on, well, it's not here, but we had we activated it with functional groups that attached on it, and then we use the char as ammonia to absorb ammonia. And what we did is we activated it and we introduced it into wastewater. And what happened is that if the feed, the wastewater concentration, had about 60 milligrams per liter, milligram per liter of ammonia, after introducing hydrochar, it got down to about 40, and then we took it out, introduced a new batch of hydrochar, and we reduced it to about 30 milligrams per liter. And that's when we used raw hydrochar, not the activated one. When we used the activated hydrochar, you see it had a better absorbent quality. So the water started at about 60, went down to the first round, went down to about 35 milligram per liter. And the second batch went down to less than 20, around 12 or so. And why is it important? Because that was, for example, the ministry, the regulation by INBAR regulations that water can be reused or sent to the environment. So that was the deal there. So we demonstrated, I think, to a certain extent that it can be used as an absorbent material for ammonia. To be honest, I would say that I don't know if that's all true, because ammonia it reduced, but I don't know how much TOC it introduced, and it introduced also some color to the water. So if we started with something which is transparent, now we had something which is brownish, but maybe it is good for gas with ammonia, okay, for moist gas or you know, things that coming from chicken coops and so on. So that's something to look at. Then we used it as soil amendment. You see here the sand. Here, when the sand is added with this char, it's black, more black. These things, these are the lettuce, and this sampler for greenhouse gas emission. And we follow the you know, the growth and what we different treatment. I don't want to bother you with the details because I don't think it's important, but the negative control, when we had very little very little nutrient solution given to the to these lettuces, something like 10 or 15 milligram per liter as a liquid fertilizer, that's what it grew. And when we had the hydrochar or the activated hydrochar or urea in hydrochar or just urea as a positive control, we see that it all grew, some better than others, that it just came 2 days ago from the experiment. I don't know even to analyze it very well. But what we notice is that there is no inhibiting features that we expected to have from the sludge, and that was surprising and really interesting, and it coincide with another study that we have on chicken manure. When we use human excretion, you'll see in a minute, we do have some inhibition. So the soil amendment using hydrochar resulted in significantly higher yield. So it definitely can be used as some sort of a soil amendment. And when we look at gases, and again, I don't want to get in too much into the details, we see some greenhouse gas emission coming out from that, and we need to figure out what it means. But basically, you can see that the ammonia, for example, that was the activated the soil amendment that had activated hydrochar that was coated with ammonia had very little, for example, emissions, in this case of N2O, versus, I don't know, just hydrochar on its own that has much higher emissions. We need to figure out what was happening there. And this is for CO2, and actually we got pretty much the same type, the same trends, obviously different numbers. But there's definitely microbial activity and soil respiration in the soil, which is letting us know that it's not toxic by no means. The other thing which is considered a burden with this usage is the liquid phase. What do you do with that? And we did the same thing. We reduced the liquid phase that contained typically 5,000, 6,000 milligram per liter of nitrogen to about 80 milligram per liter of nitrogen, which is commonly used for fertilization. And we had the negative control, and we followed the plant growth, and we actually got the same type of thing. The negative control here, that's dry weight of the lettuce. Negative control was very little, and then all the aqueous phase amended liquid grew really well, and that's the positive control when we use a commercial fertilizer. So you can see that pretty much it grows well, and it can substitute a commercial fertilizer, at least to a certain extent. The same thing was done with algae. So not only lettuce, and we used the aqueous phase as a fertilizer, and we actually had a really nice biomass growth. The nice thing about the algae is that if you start with this soil, aqueous phase amendment, which is very brownish, and when you grow algae, the water is green. But once you harvest the algae, the algae also takes the color out of this solution, and basically it ends up with being a clean water, which means that we can use the aqueous phase as a fertilizer, we can harvest the algae, and then we can use the clean water to whatever purpose that we want, obviously not drinking, but for other reuse, washing or other irrigation, but now without phosphorus and nitrogen in it, or with very little. Okay, I hope that's clear. And that's the real result. So that's the biomass. Here is a chlorella species and another species of algae, and that was the HTC aqueous phase as a fertilizer, and you see the growth, and that's the MBG11, or basically a commercial nutrient solution as the growth factor, or the fertilizer, and we can see the same type of growth if you use the aqueous phase and the commercial nutrient, and you see how the color changed from the aqueous phase will be before use, and that's after the treatment with the algae. And here I want to for a couple of minutes, just say that it's not always, as my daughter say, parparim vefeyot. There are some inhibiting properties, at least to some of the substrates, and that's human excretion. And you can see that the plant didn't grow well, and that's after irrigation with aquaspace at 80 milligrams per liter. It was lower than the negative control. And we can see the same thing if we put this aquaspace on a plate with bacteria. We can see this inhibiting ring, and we follow that a little bit more closely now. But it means that we need to know what we're doing. Not only that, we thought, and I think Roy at Neve Yaar is using these kind of things now to actually spray a plant in order to remove, in order to treat for different plant diseases. So it can be also used, but we need to know what we're doing. It's not always straightforward. And to summarize this whole thing, I would just say that this hydrothermal carbonization is an attractive, I think, alternative to wastewater sludge treatment, and they can recover energy, nutrients, and water. I think I demonstrated that, at least I hope. The solid phase is contained about 3 to 5 times more energy than needed for its production. So it's actually a net energy, positive energy balance, which is unique, I think, in terms of waste treatment. All the products are sterile and can be reused one way or another, not always straightforward. And I think that at the moment, the research is at a stage that it's really about ready to go to a larger pilot scale type experiments rather than stay in the lab. And with that, there's tons of people that probably I'm doing the talking, but other people were working. So there are lots of, many of my students, different collaborators from Germany when we had this BMBF, Roy Posmanik and Bernstein. He's from the Zuckerberg Institute, and Roy is from Neve Yaar, and Ilya Gelfand, who looked at the greenhouse gas emission. He helped us with that, and more students and technicians and lots of funding agencies. And yeah, that's it. Thanks.
Questions?
Question.
For large scale treatment plants, do you think really it can be more better, let's say, than the anaerobic digestion like the Shofdana? Because it looks maybe for small scale it can work, but
I'll say two things. First, I can say yes, I can no, but the truth is I have no clue. It's something that has to be done economically, economic feasibility and so on. The evidence that it can be, there is the first, I just read an article in the what is PA? Philadelphia in the US, where the end, Pennsylvania. So the first HTC large full-scale facility in their treatment plant, in the wastewater treatment plant, was built, and they're happy about it, and they're it's working. I know there is one company in Germany, not for wastewater, for other things, and there is one company in Spain for organic waste. So it's coming there. It's like mushrooms now. So yes, I think it has the potential. All our calculations show that it is possible. Whether applicable in large scale, it's not me, it's beyond me, but I do think it is possible. I can say also that through this US-Israel collaboration that we have currently, we're building a pilot in the Shafdan. So we'll have some more results next year, I hope.
But I think, you know, in the Shoftan, I think it's 55 degrees anaerobic digestion, like heating these volumes to 180. I don't know.
No, it'd be very, very different because you'd have to, here with the Shaftan, you have the amount of water that you use is much larger. You use usually 1 to 8, 1 to 10. The solid here, you use 1 to 2 or 1 to 3. It'd be 1/3. I don't know. It's coming, whatever comes from a belt. You'd use it directly. I have a feeling that it can work. I'm pretty sure it will, but it's not that I know that. Okay. Sure.
One is you touched actually issue. How stable is with the hydrochar in contact with water? What is the level of DOC concentration release? You know, you said there's a cola appearing, so there is certain release. So
right.
How much is released?
So it depends what, and it depends if you shake it in the water, or if you
like 1 to 10, just order of magnitude.
So what we saw that it is stable. Let's put it that way, and it is used as in terms of nitrogen that we followed more closely. We can say that it is similar to a slow release fertilizer. It's not releasing it directly, and it takes some microbial degradation to break. You know, we have 1,000 milligram per liter of ammonia, but we have 3 or 4% nitrogen in this hydrochar. So it's not the same order of magnitude even. So it released over time, that's for sure. There is some release of organic carbon, which is direct. I don't know to say, I don't remember the numbers, and we haven't followed it closely, which is something we should do. And with phosphorus, it's the same with nitrogen. It released very slowly, but it is released. So that's what I can say. And colors is probably from the organic matter that is released, and I haven't followed that closely.
Also, one technical question. You had shown data on thermogravimetry and calorimetry. That you have instrument for measuring bomb.
Yes.
TGA.
So we have TGA and we have TGA with FTIR. So we actually, I didn't show these results, but we also saw the gases that are emitted from the this after TGA. We use bomb calorimeter, and we use the equations from CHN from the elemental analysis, and we actually compare it in pretty much the same, but we always use bomb calorimeter. Not in Sdeboker, we send it. But we did send it to Ramat Chovav, and we use the yes. Can.
Eddie is asking on the chat box, are there environmental issues regarding use of hydrocharcoal for energy?
So I don't think there are rules at the moment, but the environmental issues can be gas emission. And we looked at it, I haven't showed it, but it's basically pretty much like coal. Now, people would say, if it's like coal and coal is contaminating, you know, why do you replace something bad with something bad or similar? And I would claim here that the time that coal will be disappear from the world will be years from now. But not only that, this is a renewable source of energy. We don't excavate the coal. We don't. We really recycle the waste, and this waste is already out there. So if we land spread it, it will waste the same way it is wasting with that. So we might as well use it for energy instead of using coal for energy. So that would be my answer to that.
Actually, I understand that the best thing is not creation of new source of energy. It's carbon sequestration.
Absolutely.
Prevented from degradation.
Exactly.
Multiple uses, regardless to coal use.
That's right.
You have a stable material staying in soil, floating and growing.
Yes.
Not produce CO2.
Right. But then the issues of economy, you're right. But then the issues of economy and using it for energy source would be order of magnitude higher than in terms of return to use it as a soil amendment, for example.
Just to bury it?
Not to
to use it as a soil amendment. Yes.
the field will be
Right.
sludge, gold, whatever.
Yeah.
So,
You know, when it's sterile and you have removed most of the micropollutants, it's really attractive.
another question, is it possible to compare like hydrochar with other carbon char in terms of cation exchange capacity or anion exchange capacity? Again, in numbers, how big is it?
Yes, so actually the surface area is not big like activated carbon. With biochar, it's not exactly the same. Biochar, it depends how you prepare it, but you can have a little bit higher. If you activate the hydrochar, it's becoming better than normal biochar in terms of absorption capacity, at least for ammonia. And not only in a clear solution, but also when we use wastewater, real wastewater, so with the competing ions. But it's 20 grams per kilogram. I mean, it's really, it's low. I can't remember the numbers now, but it's not, it's like 2 orders of magnitude smaller than regular activated sludge, granular activated sludge in terms of surface area. What did I say?
Carbon.
Activated carbon. I don't know what I said. But sorry, when you compare it to granular activated carbon, and but we haven't tried to activate it the same way we activate carbon. We activate it in simpler or cheaper ways because we figured that if you do that and then you activate it, you never know what you get. So we haven't done.
עמית, אפשר לשאול שאלה? זה פנחס.
The molecular structure of the hydrochar compared to biochar. In biochar, you compress the rings to flat sheets. And what does happening when you press?
When you compress it, you saw actually what it looks like. These granules, but in terms of its the molecular level, well, we haven't followed. Well, it's like carbon dots. That's how they use carbon dots. They use HTC actually to produce it, but from glucose or from pure materials. So the structure is fairly known, but it's different. It's different than biochar. I cannot say exactly. I don't remember the exact differences, but the structure is different than hydrochar.
Still have relatively higher amount of hydrogen and oxygen. If I looked at the ratios of H2C and O2C, you still, I mean, if you want to say that you are doing carbon sequestration, you need to get below 0.7, I think, in the ratio, okay? Here you show something like.
But since still you capture the carbon, then it's a carbon sequestration.
But still there is a degradation, and there is a model that says that they decided about this threshold, okay, of
I would not, I don't know, I cannot comment on that.
ratio between hydrogen and carbon.
I'm not familiar with that, and but just I'm going to say something that I don't know.
That's the reason that I asked how does it look like on the molecular base.
So it's not only chains, it also has some aromatic, aromatic compounds in it, and it's different than biochar in that regard. Yeah. Okay, thank you. יותר עדיף ככה.
הספיקר לא הרבה. תבדוק מה קורה.